This is a short animation to talk you through the function of normal lymphoid cells. So, just a reminder, in the bone marrow are hematopoietic stem cells which can give rise to any blood cell. In this animation, we shall be looking at the lymphoid progenitor cell and the cells that arise from that. So, within the bone marrow are lymphoid progenitor cells. These can give rise to naive B cells and naive T cells. The bone marrow forms part of the primary immune system along with the thymus. We'll look at T cells first. The naive T cell can differentiate into either CD8 positive T cells or CD4 positive T cells. All this means is that the co-receptors to these T cell receptors are CD8 positive or CD4 positive. Now let's focus on CD8 positive naive T cells. The naive CD8 positive T cells are activated by an infected phagocyte like a macrophage. The infected phagocyte presents the antigen on an MHC1 molecule. This is a major histocompatibility complex type 1. This process activates the naive T cell into a cytotoxic T cell, whose function is to destroy other infected cells presenting the same antigen. Okay, so going back to this picture, let's now look at CD4 positive naive T cells. Just like CD8 positive T cells, they are activated by phagocytes. The difference here, however, is that the phagocytes are not infected and the antigen is presented on an MHC type 2 molecule. The naive CD4 positive T cells activate into T helper cells. The function of these cells is to enhance the immune response by activating B cells and phagocytes. Let's now look at the other arm of this diagram, the naive B cells. Like T cells, they arrive from lymphoid progenitor cells in the bone marrow. Here, the naive B cell undergoes a process called VDJ recombination. This is also known as somatic recombination. This enables the naive B cell to produce antibodies, mainly IgM, so that it can quickly respond to an invading pathogen. The B cell then travels to the lymph node. This forms part of the secondary immune system. Within the centre of the lymph node is a germinal centre where most of the actions occur. The naive B cell recognises an antigen and engulfs it. It then processes it and presents it to a T helper cell on an MHC2 molecule. These processes result in an activated B cell. This B cell can either become a plasma cell, which secretes specific antibodies to an invading pathogen, or a B memory cell that remembers the antigen to make the antibody if the same infection occurs in the future. Here's a summary slide for you to print off. The link is provided below. If you found any of this difficult, please go back and rewatch the sections. You can pause, fast forward, rewind at the pace you want.